Hello, in this video I'm going to briefly look at DNA libraries. When working in a lab you can use two types of DNA libraries: genomic libraries, referred to as gDNA libraries, and complementary or cDNA libraries. And while both consist of DNA sequences cloned into vectors, they are different. A gDNA library is derived from genomic DNA and will contain all the DNA required to make an organism. A cDNA library is made from the messenger RNA present in a cell, and therefore the contents of a cDNA library is cell type, time of day and cell condition dependent. That is, the contents is dependent upon the messenger RNA that the cell is making at the time the library was made. So, how are the two library types made? Well, in the case of gDNA libraries, you would extract the genomic DNA from cells, cut it with a restriction enzyme to produce fragments that are then cloned into plasmids and then get the plasmids into bacteria. For a cDNA library, you would collect and lyse the cells and purify the messenger RNA from the lysate using affinity chromatography. That is, you would use oligo-DT beads to bind the poly-A tail of the messenger RNA. This first step can be tricky as lysing the cells releases a lot of enzymes that will rapidly degrade the messenger RNA. Plus, there are RNases in the environment that can also degrade the messenger RNA. Hence, equipment and reagents have to be prepared so they are RNase free. Next, you would make a complementary DNA molecule of the messenger RNA using reverse transcriptase, and the messenger RNA would then be removed using RNase H. Finally, before cloning the DNA into a plasmid and getting the plasmid in bacteria, the single-stranded DNA would be made double-stranded using a DNA polymerase. If cDNA is so challenging to make and is so dependent upon the messenger RNA expressed by the cell, then why don't we always use a gDNA library? Well, a gDNA library also has some disadvantages. For example, assuming we are in eukaryotes, a gDNA library will contain introns and exons. So if we want to capture a piece of DNA in the lab to produce a protein or work out the messenger RNA sequence for a protein, then a gDNA library will not work. Plus, unlike in a cDNA library where the gene has been transcribed to messenger RNA which has then been used to make the library, the gene in a gDNA library will most probably be spread over several clones due to how the library was made. Also, the clones may not have sufficient overlap to allow the gene to be reconstructed through sequence alignments. Okay, so you must understand the difference between the two library types, as you would need to use the right type when working in the lab. For example, there would be no point in using a gDNA library if you wanted to snag the full sequence of a messenger RNA or express the protein. Likewise, there would be no point in using a cDNA library if you wanted to work on the gene structure and its regulation. Okay, so that is pretty much all I want to say on libraries. Bye for now.